In this video, we will show you how to effectively wash plant roots from soil samples.
Root washing is an important step to accurately measure root length, root diameter and root biomass to gain an understanding of how well a plant's roots have performed in the soil environment.
The soil samples to be washed can come from multiple sources, including soil cores collected from field trials or samples from rhizotrons or long tube experiments in controlled conditions. The samples here are from soil cores collected from a field trial. In step 1, the samples are processed in the root washer. The root washer consists of 4 buckets built into a table that sits in a large open-top cylindrical tank. Each bucket has a central overflow downpipe and 2 jets which feed water into the buckets, either from a main supply or from a pump which circulates the water within the tank. Under each bucket is a funnel with a mounted 550 micron filter which captures the roots being washed out of the soil samples. In step 2, samples are placed into the root washer and washed for around 15 minutes. Depending on the soil texture of the samples, it can be helpful to soak the soil before being processed. During the washing, it may be necessary to break the soil samples up by hand to ensure roots are effectively captured. Samples are collected by washing the filters into pre-labeled containers. After this, the soil is washed for another 5 to 10 minutes or until the researcher is satisfied no more roots are appearing in the filters. Before samples are removed from the filters, the buckets are checked to ensure no roots are floating on the surface or on the sides of the bucket. The water is removed from the buckets through the filters to ensure all roots are captured. Roots are stored in water in labelled containers in a fridge and processed within 24 hours. In step 3, each sample is then processed for scanning by placing in some water on a shallow tray to separate roots from any crop debris and non-root material. Roots are then placed onto a clear tray and separated from one another before being scanned using a flatbed scanner to create an image of all the roots washed from the original soil sample. In step 4, the scanned image is analysed using the software WinRhizo, which calculates the total root length in the sample, the diameter of the roots and the length of roots in different diameter classes. The root length density of the crop in the specific soil depth sampled can be calculated using the total root length and the volume of soil sampled. In step 5, the samples are placed onto filter paper after being scanned and the water is vacuumed out. These samples are then placed on foil trays before being dried in the oven and recording the dry weight. This measure will give an understanding of the root biomass within the different soil depths sampled.